The cell membrane is a really complex organelle. And I think sometimes people forget that the cell membrane's an organelle in the same way that they forget that your skin is an organ. In fact, the cell membrane is so complex that I have another mini lecture to tell you about it. Before moving into this mini lecture, all you really need to know is that a cell membrane separates the inside of a cell from an external environment. And as we've already seen in this class, cell membranes love to take up that pinkish purplish stain, allowing us to see the cell membrane very clearly, as we see in this picture of adipose tissue. The cytosol is the liquid that is on the inside of the cell. Some of you might be saying, wait, I thought that was the intracellular fluid, or the ICF. Yeah, kind of. If we're talking about one cell, then we would call the fluid on the inside of the cell the cytosol. But if we were talking about all the cells in your body, or all the cells in a tissue, then I would use the term intracellular fluid. Some of you might also be asking, are there cells with no cytosol? It's not like we absolutely know that there's no cells with cytosol, but right now we've not discovered any of them in life as we know it. So dissolved in the cytosol are tons of negatively charged proteins, lots of amino acids, tons of little pieces of DNA and RNA, and a whole lot of potassium cations. Students often confuse the term cytosol with cytoplasm. The term cytoplasm, it refers to the cytosol inside the cell, but it also includes the organelles that are inside the cell. So in this class, we usually are referring to everything inside of the cell, organelles and cytosol, and it's usually a good idea, like if you're not sure what term to use cytosol. I'm sorry. Obviously, I don't know what term to use. Use cytoplasm. Final answer. Villi and microvilli are projections of a cell membrane. These are not tails, and these are not hairs. They do not extend off of the cell membrane. They are actually projections of the cell membrane, like little hills of the cell membrane, as shown in these figures here. We will see villi and microvilli on organs that need to increase their surface area in a small space. Your small intestine needs to increase its surface area to serve your needs, your nutritional needs of all of the tissues in your body. If the small intestine were just like a hollow tube, which is what it becomes with inflammatory diseases like Crohn's and celiacs. But if it was just a hollow tube, you would have no chance of taking in the nutrition that you need at a rate fast enough to keep you alive. I've taught from various textbooks that tell you that the surface area of the lungs could cover a tennis court. And I always want to say, like, yeah, who actually did that to confirm it? But I think that the tennis court thing is just to illustrate that there's a lot of surface area in a small area in your lungs. Cilia, which is the plural of the word cilium. Cilia are little tiny hairs. These are hairs that are on cells, and these are usually in mucous membranes, among other places. Recall that mucous membranes line openings to the outside, and they have to maintain a moisture, mucousy barrier because the external environment is constantly drying them out. So cilia can sometimes be confused with flagella. However, flagella are much longer structures, kind of like a tail, and flagella are present only on one type of cell in the human body, and we'll discuss that in a moment. Cilia don't act alone. They're usually found in thousands, if not even more, on the apical surface of an epithelial cell on a mucous membrane. That's the fancy way of saying on the cells that line the opening. Cilia will beat in unison, moving the mucus in one direction. In your trachea, all of the cilia are waving from bottom to top or inferior to superior. As all of the cilia wave, the mucus moves superiorly. And although you're unaware of it, every moment you are actually swallowing mucus from your trachea down into your esophagus so that your stomach enzymes can break it apart and digest it. Cytoskeletons, they function very much like the bone skeleton of an entire human in providing support for organelles. The cytoskeleton is an internal network of different types of proteins that they're fibrous proteins. There are 3 types of these proteins, which I'm sure you can guess their difference by their names. Microtubules are larger than microfilaments, and intermediate filaments are in between the two. Many years ago, it was thought that the cytoskeleton's only purpose was to support a cell and to provide the cell shape. However, in more recent research in the 21st century, it revealed that there were groups of proteins that move substances around the cell using the cytoskeleton as like a highway or a network of pathways. Centrioles, centrosomes, cilia, cytoskeleton, and flagella. Right? All C words and then the flagella. All of those are made out of those microtubules, microfilaments, and intermediate filaments. So a centrosome is a pair of centrioles, which are barrel-shaped structures, and they are made out of microtubules. During mitosis, or during cell division, microtubules extend from the centrosomes and connect to the kinetochores of the centromeres of centrosomes. Oh my gosh, I feel like I would have to look that up myself. This connection, what it does is it then pulls sister chromatids to the opposite sides of a cell in mitosis. When we get to mitosis, we will explain these in much more detail. Right now, I think it's appropriate to just say they're used in mitosis. The flagellum is basically a tail. However, instead of moving back and forth and back and forth and back and forth, which is very energy intensive, this tail has a rotary motion, which is way more efficient. The only cell in the human body that has a flagellum is spermatozoa, which is the official name for sperm. As you can see, the flagellum is a structure that is found completely outside of the cell membrane, much like the cilia. However, both cilia and flagella are connected to something called a basal body, which is basically just a large protein like an anchor that's found in a cell membrane. Human sperm cells exposed to temperatures higher than 94 degrees Fahrenheit during development will not develop a flagellum correctly. And this is why testicles are not held within the body cavity, but are held completely external to all cavities. That external distance reduces the heat of the testicles to approximately 94 degrees Fahrenheit, or the temperature at which flagella correctly form. This is also the source of infertility for a lot of couples, resulting from the sperm donor who might wear underwear that is a little bit too tightly binding. The energy converting organelle in cells is mitochondria. Mitochondria are capable of taking oxygen and glucose, which you see over here on the left side of this picture, and then what they do is they do a series of oxidation and reduction reactions that result in carbon dioxide and water as waste products, but also a molecule called ATP. ATP is kind of like cache in the body. It's able to be used to move molecules around, move things against a transport gradient, and a whole bunch of other things that happen in a cell, making proteins, blah, blah, blah. If you think about it, though, you're going to have lots of mitochondria in the cells in your body that create contractions. So your muscles are engorged with mitochondria. But other cells in your body may not have many mitochondria because they get energy from different sources. Red blood cells are like this. They puked their mitochondria up long ago to make room for oxygen. And they actually use an amended process of fermentation to create the ATP. No matter what, though, mitochondria, they do follow the expanded surface area concept that we've mentioned a few times. Mitochondria are little bean-like structures, and they have this folded internal membrane. And what happens is the membrane is kind of like the Hoover Dam. And what it does is the mitochondria will make a concentration gradient, kind of like the Hoover Dam traps some of the water behind the dam. And then the mitochondria will release the gradient and use that gradient or moving molecules to make ATP. This is very similar to the Hoover Dam opening the locks, allowing water to flow, and converting the kinetic energy of the flowing water into the potential energy of electricity. Lysosomes are basically little tiny stomachs that float around the inside of a cell, and I mean they do a couple things, but one of their main things is they digest broken down organelles or organelles that need to be recycled. The term lyso means to split. And rumors have it that the name or the origin of the brand name Lysol came from lyso. Lysosomes originate from the Golgi thing, and they contain digestive enzymes. An interesting but upsetting disease is called Tay-Sachs, and it's a genetic disease that comes from a malfunction of these lysosomes. In Tay-Sachs disease, the lysosomes do not contain one very specific enzyme that does lipid breakdown. And we know from previous mini lectures that if you lack that enzyme, no other enzyme is going to step up and do its job. This is a genetic disease in which the individual's DNA does not contain the recipe to make this enzyme. So the lysosomes, they become engorged with lipids. And because they have the inability to break apart the lipids, right? They basically become engorged with useless molecules. And as the lysosomes fill and fill, they crowd out other organelles of the cells, and it essentially becomes fatal. Peroxisomes. These are like our little vesicles of death. They're different than lysosomes. Peroxisomes are present to break down unfamiliar substances, not recycle or break down substances that are expected to be in the cell. So a peroxisome will not break down and recycle organelles. It likes to break down things that have entered the cell, most likely by endo or pinocytosis. Macrophages or phagocytes, which are basically cells that are capable of eating little bacteria and viruses in the external environment, they use peroxisomes to basically kill a foreign substance. If you work in a laboratory setting, you will most likely use ethanol or rubbing alcohol to sterilize instruments. But you may also use very concentrated hydrogen peroxide to sterilize things. Peroxisomes, they use that concept as well. Peroxisomes are little vesicles that are full of a very concentrated hydrogen peroxide. So when the peroxisome fuses with the vessel that's been created from the endocytosis, the hydrogen peroxide quickly fills the invading substance, And in this picture that you can see here, you can see the hydrogen peroxide as little green, like little tiny green Pac-Man. And what they're doing is they are poking holes in the cell membrane of that little brown bacterium that actually has a flagellum as well. But just poking holes in a cell membrane is quite effective at causing water to move in by way of osmosis. And what it does is it causes the bacterium to shrivel.